Hello and welcome to part 3 of recombinant protein expression and purification, focusing on his tag affinity protein purification. So now we'll get to the nitty-gritty. We've talked about expression, we've talked about cell strains, we talked about how to culture the cells, break the cells, lyse them, and centrifuge them. Now we have a nice clear lysate. Time to apply that to the column so you can get a nice purification. Select the samples and fractions that are going to retain the bulk of our peak. Run on a gel and characterize to show that we have what we think we have and see how pure it actually is. So excuse me. So the basic purification steps are the basic purification modes you can use with gradient or step elution. We'll be using a step elution, meaning we're going to add one solution, then change it to another. We're not going to have a gradient shift between one and another. You can purify these are batch, and there's a great protocol on the MCC website already that talks about batch centrifugation, where you take the beads, mix them with the lysates, let them rock back and forth while spin down the beads, or just let them settle and pull what is left behind on that supernate. The idea is that the histag proteins will be bound to the beads at the bottom of the tube. Some folks will actually have magnetic beads, so beads with a bit of iron or something in them, hold a magnet up against the tube, the beads go to the side, you can literally pour it out or pipe that out your solution, your proteins on those beads. You can centrifuge these down, use a gravity column chromatography or liquid chromatography, and if you're fancy and have a nice lab, you can maybe do a fast performance liquid chromatography or FPLC. The idea is you're going to take your unclarified lysate, you're going to mix it up with the beads, you're going to spin down or use a magnet to get the beads and take the supernate away. Your proteins will be here, mix them up, add something to like imidazole to remove or decrease the affinity between or compete with the histidines binding to the nickel beads, take that solution away, and off you go. So what are some of the key issues when thinking about protein purification? One, that clarification that we've talked about in the last video. If it's at all cloudy, or if there's viscosity from large batches with DNA, you're going to get a column that is just going to be gummed up and really slow, and you're going to be upset with how long it takes to flow through, especially when you see your neighbor that had clear samples, they didn't have cloudy particular non-viscous, it's flowing nicely. They're going to be done and gone to supper before you even get a chance to see what new Netflix are out there. So you got to make sure things are clear. Loading, washing, and eluting, there's all sorts of key issues there. In terms of how you spin up or disturb the beads as you're doing these steps, we'll talk about those in a video. You want a column that's wide enough and long enough to get a good flow and have a good surface area on the top, but not so wide that everything flows through too quickly. You want to be careful how much protein you're loading on there. A good chromatographic scientist or separation specialist will talk about how you want about the top 10, 20% of the column to bind to your protein and have the rest of this column left for just separation as you're going through each of the wash and elution steps. In the protocol, it talks about how much culture, how much protein to expect, and the size column for that. We'll hit that here as well. We'll show you in the protocol where you want a certain amount of salt in your various washes and elutions to reduce the non-specific interaction. Some proteins will still bind to nickel because they have amino acids or structure that bind in the middle, and they have histidines too. They may bind maybe less tightly or less specifically, so you use a little bit of imidazole to block that. In some cases, you may need to actually not use nickel, but use a different metal. So there's the histidines, the 6 or so histidines will bind and wrap around and then ligate two metals pretty nicely. It's the imidazole ring of the histidine side group, right? And so nickel's often used because it's got a pretty good affinity, but other proteins bind. So there's other, specificity is good, but it's not great. Use zinc or cobalt, you get really good specific interaction. Very few proteins at all will bind to cobalt immobilized to column or resin beads, but the affinity is really low. So you have to have a lot of resin, and even then a histag protein doesn't bind terribly well. But it does work if you have lots of contaminating components in whatever system you're working with. All right. The rule of thumb is when you're washing a column off, the best rule of thumb is 10 column volume. So if you have a 10 mil column, one column volume is 10 mils. And so after you put on your load, you want to wash with 10 times that column volume. If it's 10 mil column, that's 100 mils of whatever wash or buffers you're using. Depending upon the size column, this can get pretty large. So you can maybe get down to 5, 6, 7, 8, but the more you wash, the better off you are. We'll show you a trick to measure proteins coming off to know when you can actually switch over. And then storage. When you're done purifying, if it's too concentrated, some of these proteins, especially some of the MDHs, will crash out. You got to pay attention to that. Dialyze, get rid of the imidazole, and don't freeze. We'll talk about all of that. A very, very simple good rule of thumb is if you're doing a liter expression to a 2-liter expression, you need 20, 25 mil column. If you're doing a 50, 100, 20, 250 mil, you need about a 2 to 5 mil column. Will take care of most of these expressors. You may need a little bit bigger column. If you go back to the first video we showed, we had a whole table of high expressors and low expressors. You can actually look up the binding capacity for these beads and make your estimate on the size column you need. So we're talking about a nickel agarose or nickel dextran or some sort of a bead or resin that has a spacer and then a way for that bead to hold on to nickel. All right. There's different ways of doing this. You can load the column. You can allow beads to settle in a batch mode, rock it for a while, let them settle, and then pull off the non-binding portion of it. A lot of times we include a small amount of imidazole to reduce weak binding. Some folks will include that right away while binding. Some will increase the nickel in a subsequent wash space. So they let everything bind and then get rid of the weak binders with a little bit of imidazole and a higher amount of salt. Again, you want to get rid of the poorly bound proteins versus the nickel proteins. But remember, this is equilibria. The more you wash, the more likely some of that nickel histidine protein is going to be bound to the column, and it's an equilibrium between bound and free. If you wash too much, you can actually wash off the nickel because it's going to be bound and off, bound and off. That's why you want a taller column where you're only binding about the top 10%. So you can do a more rigorous wash and still maintain the histidine tagged protein to the column. You wash too long, you can even strip off those proteins. Again, it's bound and free in equilibria, and there's always a little bit of free. And if it's free, it's coming off if you keep washing it, right? And then eluting. Eluting means removing whatever's bound to the beads, your histag protein, and it's done by using imidazole. Imidazole is the side group of histidine, and it competes then, this free imidazole at high concentration, competes with the histidines bound to the nickel. And as these proteins bind and on in equilibria, then the imidazole can compete with and tie up the nickel, causing the histag protein to come out of the column or elute. Once you're done with that, then you need to look at the protein concentration of your samples. Make sure if you're doing an enzyme like MDH, you want to make sure it's active, so you measure its enzymatic activity. Maybe you want to look at KMV max or specific activity to show that it is what you have. If you have mutants, then you do those kind of experiments. You do a SDS page to look at the purity. So here we have a simple gel, an image that we borrowed from Qiagen. You see here in the load, you have all sorts of proteins, including the one we've expressed. And as you wash it through, there was a little bit of leaking of our protein. And then here's in our elution, we have the protein that we're looking for and a couple of other proteins. These might be proteins that tightly bound this, or chaperone proteins, or these proteins could have bound to the nickel by itself. So this is still a pretty clean sample. If you were to look at the total, if you were to actually measure all these proteins out, or scan this and then look at quantitatively how much protein's in each band, you're 80, 90% pure. So maybe you need to run a second column in this case. But then you do this purity, you run an SDS page to look at the purity, and then you can use molecular weight markers that you have here compared against the amount of migration through this gel, you can create a standard curve where you look at the RF factor here and this RF and then estimate its molecular weight. So you make sure not just that the biggest band you're looking for, but it's the right molecular weight. So what is the basic approach for this? So here is a nice 5 mil column for 100, 200 mil culture of MDH. All right. You see the nickel column with the right color here, nice kind of a blue greenish. We have a stopcock down here and a tube ready to collect in our samples. So you want to prepare for about a liter of culture, 20, 25 mils of beads. You take 50 mils of a 50% slurry, put it in a column and wash things through. I always like to start with eluting at the, or using an elution buffer, so high amount of imidazole, in case the person that used these beads before you had left some protein on there. You want to make sure everything's washed off that can be washed off. And then you want to wash off with plenty of what we call binding buffers. So lower amounts of imidazole, a little bit more amount of salt, something that really helps the MDH histag proteins bind to these nickel beads. If you're going to go through and run a gel when you're done with this, make sure you save some of that lysate before you put it on the column. Use a little bit of that clarified lysate. You can either freeze it or you can add some SDS page sample buffer to that and then store it later on. So there's batch binding. Batch binding is when you take, you would take your beads, you would put your lysate on there, cap it off, and then rock it, let it sit straight up. Otherwise, you can just flow it straight through the entire time, which is what I'll show you here in the column binding format. So let's talk about the column purification. So here we have the binding buffer, and we use this in our MDH as a lysis buffer as well. When we're lysing the cells, that's when we add some of the beta-mercaptoethanol and PMSF. So our basic lysis and binding solution will have 50 millimolar tris at about pH 8, a small amount of imidazole, 1 millimolar imidazole, 100 millimolar sodium chloride, and 0.1 millimolar EDTA. And for doing lysis in your binding, you want to include PMSF. Once PMSF goes from the alcohol stock into the water, that PMS will also then react with the oxygen as well as the OH and the serine in the proteases. So it's got a very short half-life of about half an hour to an hour. So you add it fresh and then use it immediately. We look at the wash buffer, we see again the 50 millimolar tris at pH 8, but now we've cranked up both the sodium chloride and the imidazole. Again, PMSF, but in reality, if you're tight on budget, you can stop using PMSF at these two steps because you've washed away and inhibited all the proteases that might still be around. Use higher amounts of sodium chloride and imidazole to block off or to wash off weak and non-specific bound proteins to the nickel beads. And then when it comes time to elute, we'll use the elution buffer where we drop down the sodium chloride. So that way that's not a problem later on. But more importantly, we have about 300 millimolar imidazole, which is more than enough to elute any histag protein from a nickel bomb column. One other thing is, how do you know how far you wash? When you take your lysate and wash it through, I'll show you in the video, you take a little bit of about a half a mil or so of 1X Bradford or Biorad protein assay solution and add a couple drops, 20 drops or 20 microliters or so of what's coming through the column. If it turns blue, you got lots of protein. When it stops turning blue, you can switch over to the next column. You'll see that in this video. And here's a whole fraction elution from one of these smaller columns. So I'm going to slowly add, try to minimize the disturbing of the bead bed. If I got some of that on there, I will open up and allow the lysate to start flowing through. Very carefully, again, without disturbing the beads on the top of the bed. Continue adding the lysate. And we'll let this flow until the meniscus just reaches into the top of the beads. All right, so after about half
or less of the column volume has started flowing through, the protein that doesn't bind should have already start to elute. To test this, we're going to take a 1/2 ml of Bradford protein reagent and put 1 or 2 drops of the eluate into that. 1, 2. It's a very slow flow rate today. You can see how blue it is. In fact, if I put this against this tube, this was control, so just the buffer, 2 drops of buffer, here's 2 drops of protein. We have a lot of non-binding protein coming through there now. So I'm going to slow.
All right, so it's important when you're adding the various wash and elution buffers not to pick up a lot of the beads. You'll dilute it. This is mostly important when you're doing the elution, but certainly where you're doing the washes, because what you're doing is any of the proteins that are bound to the columns, especially in elution, will then come be mixed up into that total volume have on top of the beads. And you're really then diluting out your sample, and you're going to have a hard time getting a nice tight Gaussian curve as the proteins are coming off of your column. Let's watch a little bit more here.
Now that we've washed the non-specific protein off with the binding and wash buffers, we've put some imidazole containing elution buffer, and we're starting to collect 1 ml fractions.
One thing that I like to do is when I first add the elution buffer is I run about 1 ml or 2 into that column of this imidazole containing elution buffer, and I stop the column, and I'm kind of what's called a pulsing this, allowing the imidazole to really loosen up, if you will, or the imidazole to compete with the histag binding proteins so that they come off in a tight band as you go through.
And let's see if we have protein now eluting off. We've had about 5-6 mils come through, 2 drops, back into the fraction tube, the new one, and you can see lots of blue. Got a lot of protein coming off now, so we're in this fraction right here. This fraction had a little bit of blue. This one had a little bit more. This one's quite a bit blue. So we'll keep going until we're back down to the point where we don't see protein showing up in our Bradford reagent.
All right, so this was the 100 ml culture volume run on a 5 ml sample. I'm collecting 1.5 or a little bit more fractions along the way. If you're going to run a 500 1 liter column, you're going to want to run 15-20 mil fractions. You sure as heck don't want to run fractions of 1.5 mils, because you'll be doing hundreds of them at that point. So you got to have bigger fractions to save with the bigger columns you run. If you're not sure, ask your instructor. On the protocol handout, it gives a guide for how big the fractions should be. So which ones do you save? Well, there's lots of protein here. So these are pure proteins eluted from there, right? So this is the first, after you started putting the elution through, here's that first fraction, here's the second fraction, third fraction, fourth fraction. There is some blue here, but if I collect these 3, or maybe even just these 2, I won't get the full yield, but I'll have the highest concentration. And if you have a small, low concentration by collecting more samples, but you want a higher concentration, you have to make this choice. They're all going to be about the same purity. Some people will take this and just pool them and then process them. Other folks will run the samples and run them on a gel and then decide. There's all sorts of ways to do this. But here's an important fact. Notice I had Bradford in these samples, or in separate samples over here. These were empty tubes. Never, never, never elute directly into your Bradford containing tubes. Bradford dye and the phosphoric acid and other reagents will denature and kill your sample. You've just wasted your entire time. So which fraction should you save? The top darkest blue. You're going for concentration versus yield.
Now that we've washed the...
All right, so now that you've purified this, like I said, you can run the lysate, you can run the various fractions of this thing, the lysate, the pellet, the supernatant. You can watch the flow through. Here's protein that didn't bind and came through, and here's your elution. So you run a gel, look for the right molecular weight, look at the purity. You want to do an enzyme assay. You certainly want to do a protein assay of this. Perhaps in biochem lab, we're going to do a Western blot so we can know that just because this is the biggest band doesn't mean it's your protein. It could be contaminating. This could be your protein. A protein underneath this could be your protein. It depends on the size. But if you really want to know if this is my protein or this is my protein, you conduct a Western blot, you analyze your sample by Western blot, and then compare where the band is with that band, and you can really get a good characterization. There's a couple tricks I think it's important for folks when they're doing purifications. Some of these proteins are happy being concentrated. Watermelon MDH can be 3-4 mg per mil and stays in solution for a long time. Other proteins and some of the MDH proteins, when you get above a mg per mil, even overnight, they'll start aggregating and crashing on a solution. They'll start partially denaturing once they aggregate. And that's really frustrating to go through this entire process, come back the next day and you thought you had 10, 15 mils of purified protein and you see cloudy precipitated proteins. It's a very sad day. So sometimes what I like to do is to prevent that aggregation before I leave for the night, before I dialyze, before I just put it in my solution in the cold room or on ice, I will do a little simple plate assay. I might not even have to do the calculations, but I'll take a standard set of BSA from 0 up to, you know, a meg and a half mil of BSA, and then I'll take my samples as I purify them and pool them, and then I'll look at that and look at this sample here is about the same color of blue absorbance as this one. If this was 1 mg per mil, this was half mg per mil. All right, so I'm not above a mg per mil. I'm not going to worry about it. If my sample, the same volume that I added BSA to, the Bradford here was as dark blue or darker than this, I probably diluted out with dilution volume or binding buffer to decrease the concentration so I won't get that crash out to happen. If you got time and energy, then what you're going to want to do is dialyze. You're going to take your purified sample, your collected tubes, your fractions all put together, and you're going to put them in a dialysis tubing. This dialysis tubing is porous, and it's designed with a certain molecular weight cutoff. 500, 1,000, 10,000, 20,000, even 30,000 molecular weight cutoff. I mean, everything smaller than that molecular weight cutoff can easily go back and forth, or through diffusion and osmosis, even the concentration out. So if you use a protein like MDH, that's 32, 37 kilodaltons, using a 10,000 kilodalton or 5,000 kilodalton cutoff, that means the MDH won't go in and out, but the imidazole, the sodium chloride, tris, or whatever else you're looking at will then equilibrate between what's inside the dialysis tubing and the dialysate that you prepared. So at equilibria, which can take a few hours, oftentimes we just leave this overnight and then change and do a second one. Think of this as CV. If we started at 300 millimolar sodium or imidazole, and maybe this was 10 mils, and if this was in a liter, that means at equilibria, that imidazole is no longer in this 10 mil fraction. It's in the 1 liter plus your 10 mils of initial sample, equilibrated to equal concentration throughout. So then you've got 300 times 100 divided by 1,100 for your new imidazole concentration. If that gives you a low enough number that you're comfortable with, I like to get less than 0.1 millimolar EDTA before I'm done, then remove the media, replace it with fresh cold media, let it sit for 4 or 6 hours or another overnight to get all the imidazole out. Imidazole takes a long time and lots of washes to really get rid of the imidazole. Never freeze, certainly don't freeze with imidazole, and don't freeze the sample until you've added glycerol or other additives for stable storage. Look to your instructor for what needs to be done, or that work with your instructor. He or she will take care of making sure you add the appropriate things. Sure as heck, don't freeze your purified samples without these additions, because it will, when you thaw it out, it'll be dead, all precipitated out. A couple of examples, if we tested all sorts of different additives, glycerol, ammonium sulfate, sodium chloride with beta-mercaptoethanol, trellulose, glycine, you name it. Watermelon was pretty good at 4 degrees, so that's at the fridge temperature for a long period of time with all of these. Watermelon or human MDH stayed by itself, actually started falling out of solution after 10, 15, 20, 30 days. With glycerol and a couple other additions, it stayed in solution. We did the same kind of studies when you freeze and thaw them back out. And again, we have a stable mixture of beta ME or sodium chloride and some glycerol that works for freeze thaw versus staying at 4 degrees. So the last thing you might want to do is process your MDH. So if you're just going to do some really cool kinetics or specific activities and make some different mutants and then compare the kinetics or the regulation of this, you can leave that histidine tag on there. That's going to be fine. If you're going to do crystal structures, some folks leave the histidine tag off. A lot of people want to take it off. But maybe you're looking at protein-protein interactions or doing some other kind of protein-based experiments with it. Maybe you want to remove that histidine tag on one or more of your proteins. Well, that's okay. We've engineered our MDHs. In this case, here's our MDH1 with what it says plus TEV, which means that here's our MDH. We have a series of additional amino acids that we've coded in the gene for this, and then the histidine tag. The amino acids you see listed here are a target for a protease called TEV protease. So TEV is this protease. It'll bind to, excuse me, 1 or 2 amino acids away from the end of our MDH, cleaving off the his tag from our MDH. So TEV protease, again, TEV is a protease. It comes from a tobacco etch virus, thus the TEV or TEV. It's a cysteine specific virus, and it likes to cleave at a very specific sequence that you see here. And actually, that's not the right sequence. Here's the correct sequence for the TEV sequence, and it'll cleave between this glycine and glutamine. The idea of this is a lot of the TEV proteases are also his tag. So if you take your purified MDH, get rid of the imidazole, add TEV protease, it'll cleave, get rid of your his tag, and the TEV protease itself has a his tag. So you take this whole solution and wash it through another nickel column, the TEV protease will be washed away, as will your his tag and any uncut MDH or CS. So you can now have unlabeled, TEV-free, protease-free proteins ready to go. All right. So now we've seen expression, culture, purification, and some of the things we do after we've done the purification. I hope this helps your experiments. Good luck.